Okay, so today we'll be talking about Mendelian genetics. But if this thing lets me. Okay, so just a couple of announcements. Just one of the things I want to mention is that when you guys send me emails, please include which section you guys are in. I have 3 sections on Wednesday, so it gets a little scrambled when I'm like trying to find you guys in the middle of all my 74 students. So please, if you emailing me, just say you're in my Wednesday 4:30 p.m. section or my 4:30 p.m. section, okay? I will be closing out quiz number 1 and 2 tonight. So if you guys haven't done it so far, please do them, okay? It's not too bad, I think. Quiz number 3 will be up. And as usual, the password will be in the discussion video, okay? Okay, so I know we did not get to meet last week. That was crazy with my crazy work schedule. Do you guys have any questions? Were the videos helpful from last week?
I think the videos were helpful.
Anyone else have any issues or complaints with those? Since all of you guys have your videos off, I can't really tell by, so. So I'm gonna assume everyone was okay with everything that was last week. Sorry about that again. Okay, so one of the things I realized after putting this up, I realized there was an error in my video. So with this question, I kind of mixed up 2 different questions in one, together. So I fixed it in the PowerPoint, but I just wanted to make sure that you guys were aware of it as well. So during which stage can cells either exit or enter the cell cycle or become committed to completing the cell cycle? So that is actually G1, not the S phase. That's when it can get released out of G1 into G0, which is where cell cycle rests, okay? So that's that question. So I had a couple of students ask me for examples on like linking numbers, supercoiling, and all that. So I'm gonna have a couple of questions here. A circular bacterial DNA that is 2,000 base pairs in size has a linking number L of 100. What does this tell you about the DNA molecule? Assume that this molecule is a Watson and Crick DNA structure of 10 base pairs per turn. So what do you guys think? Guys? If you guys don't talk to me, I'm gonna have you guys all turn on your videos so I can at least see your faces if you're confused or not.
Does it mean that it has 20 turns? Like it turns 20 times?
Wait, say that again?
Does it mean that it has like 20 turns?
Okay,
I
so
don't know if that makes sense.
200.
Okay.
Yeah, so we have our linking number, right? So linking number is essentially the number of right-handed turns that one DNA strand can make around the other. So that's 100 of them. Then we have twist, which is the number of turns the double-stranded DNA has itself, okay? So in this case, it would be 2,000 base pairs divided by 10 base pairs per turn. So that gives you 200 turns for the double-stranded DNA, right? Does this make sense so far?
Yes.
Okay. Now we have something called right. Right is an expression of the number of supercoils within a DNA molecule. So in this case, this is the equation for linking number that we have. So L equals T plus W. So to calculate out right, we would make it W equals L, linking number minus twist, okay? So that would, when you calculate it out, it gives you negative 100, right? So having a right of negative 100 means that you have negative 100 negative supercoils. If this was positive, then it would be 100 positive supercoils, okay? Does that make sense?
Yeah.
Okay, great. So I guess I forgot to turn the animations on for this guy. But this is a similar question, except instead of 2,000 base pairs, you have 1,000. And when you come down, you have 100 turns per for twist. And then right, you end up with 0. Right of 0 is essentially saying that you do not have any supercoiling going on. Does that make sense?
Yeah.
Okay. So now, do you guys have any other questions before we go into presentations, like from last week's stuff? If not, we can go into presentations, whoever wants to take a start.
I can go first. I just don't know how to share my screen.
Let me just allow you guys. Okay. So can you share now?
Yeah, I think. Yeah. Okay, so my name is Shima. My question was on mitosis and meiosis, and it was how many individual different kinds of gametes can be produced by an individual with the genotype AA, BB, CC, DD, EE, FF. And the answer choices were 32, 16, 4, 8, and 64. So the background information that we have is that we know the formula to calculate the number of gametes is G equals 2 to the N. N would represent the number of heterozygous pairs, and G is the number of gametes. So in this specific question, we see that there are 3 heterozygous pairs. It's the B, the E, and the F because they have both an upper and a lowercase letter. That would make the equation G equals 2 to the third, which would make G 8, meaning there are 8 gametes, and B would be our correct answer.
Good. Does everyone understand what we just did?
Yes.
Yeah.
Yeah.
Okay. Who wants to go next?
I could go next.
Okay.
So, can you see my screen?
Yes.
Okay. So my question was, if Avery, MacLeod, and McCarty had determined that the transforming molecule was a protein, what experimental results would not have been observed? So A, heat kill cultures treated with proteins would have transformed the R cells. B, heat kill cultures treated with proteins would not transform the R cells. And C, heat kill cultures treated with DNAs would not have transformed the R cells. And D, heat kill cultures treated with RNAs would not have transformed the R cells. So some background information. Avery, MacLeod, and McCarty experiment reported that they found the transforming principle to be like DNA beyond reasonable doubt. They took virulent smooth strain, they heat killed it, homogenized it, and filtered it. They took the filtrate and added RNAs to one sample, proteins to one, and DNAs to the third. They then added treated samples to a living, non-virulent, like rough strain. The RNAs and protein samples contained the transformed bacteria, the smooth, while the DNAs was the only culture that didn't. So this showed that DNAs destroyed the transforming substance. And then I just found this like picture from the textbook that I think gave like a better description. So it shows that they started the isolation procedure with a quantity of around 50 to 75 liters of the liquid cultures of type 3S virulent cells. The cells were centrifuged, collected, and heat killed. And then after homogenization, they extracted like carbohydrates, lipids, and proteins. Through this, they were able to obtain a soluble filtrate that retained the ability to induce transformation of the type 2R avirulent cells. They then treated it with protease, ribonuclease, and deoxyribonuclease in order to like eliminate any probable contaminants from the final product. And through this, they were able to find that like DNA was the active factor in transformation. So for my answer to this question, I chose B. Heat kill cultures treated with protease would have not transformed the R cells. I chose this because if they found protein to be the transforming molecule, then they wouldn't have observed protease not transform the R cells. Protease would have transformed the R cells if it was the transforming molecules. So yeah. And that's it.
Yep, that is correct. Everyone good with that question?
Yep.
Great. Okay. Who's next?
I can go. I was number 3.
Okay.
Um, Can you guys see my screen?
No.
I'm sorry, it's sending me to my system preferences. I don't know how to share it. Oh, I see. Can you see that?
Yes. Okay.
So it says September 9th. I was supposed to present last week.
That's okay. That's my issue.
So my question was number 3, and it was, what conclusion could be drawn if Hershey and Chase had found only S35 in their bacterial cells? And these were the options. A says protein is the genetic material. B is DNA is the genetic material. C is DNA and protein together serve as the genetic material. Or D, no conclusion. And so in order to find this answer, you have to look at the blender experiment from Hershey and Chase. But if you go back, Frederick Griffith was the first scientist who actually found out that genetic material can be passed on. And in order to find out what that genetic material was, 3 other scientists, Avery, MacLeod, and McCarty, they concluded that DNA was a genetic material through their enzyme experiment, which is basically what I think, I forgot her name, but she just explained it. I think it was Megan. So in that experiment, they used the enzymes protease and the other 2 for proteins, DNA and RNA, and that's where they got their conclusion that DNA was the genetic material, because it was the only one that, even though the enzymes broke it down, it still showed up. And then Hershey and Chase, they were also scientists, but they were also skeptical about the DNA being the genetic material. They believed that it was protein. So they did the blender experiment. And in the blender experiment, they had radioactive phosphate, which is P32, and they added that to the, oh wait, no, but they also had the radioactive sulfur, which is S35, and they added it to unlabeled bacteria. And after that, they placed it in the blender in order to remove the phage and detach it from the bacteria itself. And when they did that, they saw that the protein, the sulfur, it wasn't showing up in the bacteria. It was unlabeled, but the DNA, it did show up, and the bacteria cells, they were radioactive with the phosphate. So their conclusion was DNA was the genetic material. So my question, when you go back, it says, what would have happened if they only found the sulfur? And since they were only comparing DNA and protein, my conclusion is that it would have been protein.
Good. Who wants to go next? We have about 2 more presentations.
I'll go next.
Okay.
Sorry, just give me a second. Okay, so I had a pretty simple question, I think. It was, if a typical somatic cell has 64 chromosomes, how many chromosomes are expected in each gamete of that organism? I said in order to solve this question, you need to understand that gametes contain half the amount of cells that are found in diploid cells. We all know that human diploid cells have 46 chromosomes, and human haploid gametes have 23, which is half. So I think it's important to note that during the process of meiosis, the cells are reduced in half because of cell division. So that's how I got the correct answer is 32, because once you divide 64 by 2, that's what you get.
Yep. Great job. Okay. Megan.
Okay, I can go next. Okay. Can you see my screen?
Yes.
Okay. So my question was, what is the outcome of synapsis, which happens in meiosis? So some background information on this. So this happens in prophase 1 of meiosis. So I just have like a little figure here that goes over what happens in prophase 1. And I have the important part that we really want to look at circled here, which is what's going on with these chromosomes. And what's happening here is that we see these homologous chromosomes coming together, and eventually they will, at some points, attach to each other, and as we know, continue on into crossing over. But the important thing that we need to see here is just that the homologous chromosomes are coming together and aligning side by side. So that's how I get the answer D, side by side alignment of homologous chromosomes.
Good. Everyone okay with the presentation questions?
Yep.
Okay, so once you stop sharing, Megan, we can return back. Okay, great. Okay, if I can find my PowerPoint, that would help. Okay. So any other questions before we move on to more Mendelian genetics stuff? Yes, no, maybe so.
I'm okay.
Great, okay. So what are the Mendelian laws of inheritance? What's going on with Mendel's laws or postulates? Do you guys remember?
One of them was that he said that unit factors exist in pairs.
Mm-hmm. What about the others? Anyone have an idea?
He also talked about dominant and recessive
Mm-hmm.
genes. So when a dominant unit is present, the recessive one does not show.
Yep. What else? There's two more.
He also talked about segregation. So
Mm-hmm.
the paired unit factors separate during gamete formation.
Good. Okay, so oh. Did someone say something? No? Okay. So I put down these three in here. So law of segregation involves the alleles of the same gene separating out from each other. Okay, so the gametes end up carrying only one allele. Law of independent assortment, on the other hand, is genes of different traits or different genes, okay? Alleles. Does that make sense?
Yep.
And obviously we know that these alleles come in pairs because we have homologous chromosomes, one from each parent. And one of the other things is law of dominance. So in this case with Mendel, he saw that some alleles were dominant while others were recessive. So an organism with at least one dominant allele will display the effect of that dominant allele, okay? We cool with the postulates?
Yep.
Oh, yes, and they do exist in pairs. Okay, this thing doesn't, okay. So I don't think you guys got to this in lecture, but so the product rule for, so there are two types of probability rules that you can use to predict genotypic and phenotypic outcomes. So with the product rule, it states that the probability of independent events both occurring is the of independent events both occurring together is the product of their individual probabilities. So so for an example, if you roll 2 dice, the outcome on one die is independent from the other. So if you want to roll a 4 it's 1/6, right? You have 6 sides. It's 1 side out of the 6. If you want to do 2 4s, that's independent of each other, right? So in this case, you would be saying, 1/6 times 1/6, right? Because it's 2 different dice at that point, right? Next, we have what's called the sum rule. Sum rule states that the probability of either of 2 mutually exclusive events occurring is the sum of their individual probabilities. So for an example, if we're looking at the probability of rolling 2 4s or 2 3s. So to do that, we know, okay, so for 2 4s, we know that it's 136, right? But you're also checking, like, it's like an either or kind of situation. So we kind of use the sum rule for this. So that's how you would end up with 1/18 for this condition that you have. Does that make somewhat sense?
Yeah.
Okay.
Yeah.
So an example for this is in a family with 2 children, what is the probability of there being 2 females? So how would you guys go about this? So we have our Punnett square. Female, male, our crosses come out. So we have yeah, so for 1 female, you'd have half, and then 2, you would multiply half times half, right? So it would be 1/4 of a chance if you to get 2 females in one go, right? Make sense?
Yeah.
Okay, so now we're gonna look at 2 females or 2 males, okay? Nope. So in this case, you would add them because you're kind of having an either or situation. So for 2 females, it's 1/4. Same thing for males, it's 1/4. But when you're looking at either of them, you end up with half, right? Okay, so next we have 1 female and 1 male. So in this case, how would you guys go about it? Nobody have a clue? Okay, so in this case, order comes into matter, because you can have either the first one being male, second one being female, or the first child being female and the second one being male. So in this case, in both cases, you come up with this ratio, right? So there's half a chance of being a male in the first child, half a chance being the second, and then same thing goes for the second set, right? So we have these 2 combinations of those guys, and we end up with these 2 probabilities for each set, right? So when we try to figure out the probability of this kind of scenario, you would add those 2 probabilities together and get 1/2 as your answer. Does that make sense?
Yeah.
Okay, great. So monohybrid crosses, what's going on there? What are we looking at when we look at that? Maybe. Okay. So a monohybrid cross is mating between two individuals who have different alleles at one single locus. So like an example would be like this. Okay, so if you have, this is heterozygous, we are having a heterozygous here. So when you cross them, that's what a monohybrid cross looks like, okay? So in this case, you have the probability of having a dominant homozygous trait would be half times half, which gives you 1/4, right? Because you get half a chance from this parent, half a chance from that parent, right? Next, it's the same thing with the homozygous recessive genotype. You have that same ratio there, which gives you 1/4 of a chance for that. And then with heterozygous, you do a similar thing with the way we did the siblings situation, right? Earlier when we saw, because like this can be in any order, right? Because you can get a big P from the dad, little p from the mom, or little p from dad, big P from mom. So at the end of the day, it's the same thing. However, you got to account for both of them. So that would be half a chance. This is within a monohybrid cross, okay? So essentially you have 3/4 being purple. So that's how we see the law of dominance, right? So the heterozygous ends up purple because you already have a dominant allele, okay? And then with the recessive guy, you see it as 1/4 for white. So that's kind of, this is the genotypic ratio. It's 1 to 2 to 1. So 1, 2 heterozygous, and then 1 homozygous recessive. Are we cool with monohybrid crosses? Like the concepts behind them and like how you can actually calculate out the probability of that child, I guess?
Yeah.
Great. Okay. So dihybrid cross is essentially like a monohybrid cross, but you're looking at 2 different genes, okay? So in this case, that is essentially what I have written down. It's looking at it for 2 different traits. This is the cross that allows us to visualize the law of independent assortment, that the inheritance of one locus has no influence over the inheritance of another, okay? And these crosses, they typically show a 9:3 to 3:1 ratio in genotypically. There will be some cases where it can have modified ratios. We won't talk about that today. Just know that this is the ratio when we're talking Mendelian genetics dihybrid crosses, okay? For at least this unit, because that's epistasis and everything else, that's next week's problem. Okay. So Mendel crossed peas having round yellow seeds with peas having wrinkled green seeds. All F1 had seeds that were round and yellow. Predict the results of test crossing these F1 plants. So what do we know from this question? Anybody got a clue? So what do we know, looking at this question, what do we know about the parent peas?
One's dominant, the other's recessive, and the offspring are all the same phenotype.
Okay. So we're gonna label these guys down. Oh, okay, my animations are backwards today. Okay, so when we look at this, so we're gonna name these guys round. Ones are gonna be dominant because we have all of them having the same one. And then yellow as dominant as well. Wrinkled and green would be the recessive ones. So we know that these guys are heterozygous, right? Oh, wait, no, hold on, sorry, my bad. Okay, so we know that the parents have either so it's either homozygous for both of them, right? Because in this case, you see only round and yellow characteristics. So you know that these guys are dominant and these guys are recessive. So you end up with a genotype for this guy, right? So it should be that, but to actually understand how this kind of occurs, to make sure that it is actually happening that way, is that you can test cross. So test crossing is actually a way to figure out what the zygosity of an individual is, whether it's homozygous or heterozygous, okay? So in this case, so to do a test cross, it's essentially doing one of these crosses with the with this possibility, with this, the homozygous recessive for both traits. So I do it this way to leave it a lot less complicated. So when we do this with big R, little r crossed with little r, you get half round, half wrinkled, right? Then you look at this guy with big I, big Y, and little y, crossed with little y, little y. So you get half yellow, half green, right? Because of the what we label them as, okay? So for getting the probability of these guys, it would be, so round and yellow, half times half equals 1/4. Round and green is still half and half, which is equal to 1/4 again. So that's how you kind of go through these questions. Does that make sense? Are we okay?
Yeah.
Or, okay. So this is another question that involves test crossing. If you had a fruit fly that was of phenotype A, what test would you make to determine if the fly's genotype was big A, or big A, little a? What do you guys think? We would do a test cross, right?
Yeah.
Yep. So we do a test cross to little a. If the fly was big A, all the progeny will be phenotypically big A, right? It would be dominant. that's how it would come out because it would end up with heterozygous offspring, right? If it was big A, little a, then we will have half of them being in heterozygous or dominant A, or we'll have half recessive, right? Because that's what you get from each parent. Does this question make sense?
Yeah.
Okay, great. Okay, so this is a question, which types of phenotypic ratios are likely to occur in crosses when dealing with two gene pairs for which all the genotypic combinations are of equal viability? Do you guys remember?
Is it A?
Yes. So whenever you guys see two gene pairs, always think dihybrid, which is this ratio, okay? So next we have human blood types. I don't know if you cover this in this chapter or next chapter, but this is one of the examples of like dominant versus recessive. So in this case, A and B are always dominant alleles, and O is recessive, okay? So if you have A and B together, they both show themselves. Does that make sense? So it's like these donuts. Okay, so if you have type A blood group, you'll have all the A antigens going on. If you have type B, you'll show up with all type B phenotypes, antigens. And then if you have an AB blood group, you have A and B showing up because they're both dominant, right? So none of them mask each other. Then you have type O, which doesn't show any antigens. And A and B are always dominant over type O. So we have a child with a blood group AB and a mother that is also AB. What are the possible blood types for the father? What do you guys think?
It could be A or AB, or just AB because only AB shows up.
Okay, so it can be any of the three because it could just be the child could be getting A from mom or B from mom. It can be either or for that too. And or the father could be AB and they're just getting one of each from whichever parent. Does that make sense?
Yes.
Okay. So blood types that would exclude a male from being this child's father. What do you guys think?
O.
Yep. So if you had a child and both parents are O, don't expect a, that's not happening because O is always recessive to these guys. So with A, you can have A and O alleles. With B, you can have homozygous for A or heterozygous for A as well with an allele for O, but only A would show. Same thing goes for B. Does that make sense?
Yep.
Okay. in a disputed parentage case, a child, oh, okay, this is pretty much the same question reworded, but child's blood type is AB, while mother's blood type is AB. What is the possibility of the father? Same as before, right? So we have B, A, and AB, right? So I know you guys haven't gotten into this part yet in lecture. So I'm gonna go through this gently as possible because I don't want you guys to get freaked out just looking at the word binomial theorem. So essentially, this is another way to calculate the variability, like the probability of alternate ways to achieve combination of events. So in this case, you have this theorem, which is A plus B to the nth power equals 1, where A and B are the probabilities of two alternative outcomes. So this theorem works only with two different outcomes, right? Because it's binomial, right? And N is the number of trials. So to work with this guy, I definitely have another video for this guy, but essentially this is how you would expand them out. If you guys need a more in-depth explanation, I also have a video on Blackboard. I will open that up to you guys too. But for now, just don't worry, okay? So this is kind of the expansion of each of them up to the power of 6. This shows it like that. These are the coefficients. When you look at these guys, when you start with A and go through, you kind of go down from 3 to 1, like the exponents, I mean. So when you decrease the exponent, the other term here gains an exponent. Does that make sense? Somewhat? Guys?
Yes.
Yeah.
I don't know. A
Okay.
little confused.
Oh, so where am I losing you?
I don't know what that has to do with the genetics.
Which is where I'm getting to, actually. So this is an example of a question that comes out of binomial theorem, because so you're kind of looking at why, what is the probability of a family of 5 children that kind of ends up with, okay, so this question, I think that will answer your question as to why this is relevant. What is the probability in a family of 5 children that 2 will be male and 3 will be female? So if you try to write this out with the different number of orders, you'll go nuts. So instead of trying to pull all your hair out, we have 2 different outcomes, right? So the probability of having a male is a half. That is your A. You can put it either way. I just put it like this for now. A equals probability of male, which is a half. We have B, which is equal to probability of the female, which is equal to a half as well. And then you have 5 trials, or 5 children in this case, right? So what are we looking for? We're looking for the probability that in the 5 children that they have, 2 will be male and 3 will be female, right? So we go back to our chart, we find the one that has n equals 5. We take that expansion and then we look at, so these numbers here, 2 and 3, become the exponents for A and B, okay? So in this case, we would be looking at, oh, no, go back. Okay, so we will have 2 males and 3 females, right? So this term here, we take that and then we plug in our probability values. And that is the probability that you would get if for the family of 5 children with 2 males and 3 females. So it's just a simpler way to calculate out the probability for this, instead of going through every combination that it could be. Does that make sense?
How are the exponents used for the number of offspring that are male or female?
Okay, so these numbers, the probability for one male is a half. So you're putting it to the number of males that you're looking for. That allows you to figure out that. But then there are like 10 different combinations that you can put all of this together, right? Does that make sense?
There are 10 because there are 10 children in total?
There's like 10 different combinations that you can do this.
Okay.
Are we all cool? I know this is like information overload at this point. Yes, no, maybe so.
That sounds good.
Okay. So chi-square test, this is essentially allowing you to find the probability of a chance deviation from the expected ratio. So it's the difference of the observed the difference between the observed and expected values squared over the expected, okay? And you sum that. So essentially, you're looking at how likely it is that this observation is actually due to chance, or how much of a deviation you see in this case. So an example would be this guy. So we have a sample size of 200, right? So this is the various genotypes that you have. So in this case, you realize that, okay, so these numbers are close to a 1-to-1 value. However, we're going to use the chi-square test to see how much of a chance this deviation is from the magnitude of a 1-to-1 ratio. So that's our expected values. So in this case, since we have 200 progeny, we will take a probability of a 50 to 50 to 50 to 50 ratio, because it's 200 divided by 4, right? The total number of progeny divided by 4, because you are looking at a 1-to-1 ratio with all 4 of these genotypic categories, okay? So the chi-square analysis in this case is testing a hypothesis that there is no significant difference between the measured values and predicted values. So in this case, okay, so this is pretty much calculating them all out, like take them step by step because you don't want to mess up with the calculation itself. So these are values that you would get with these guys. The chi-square value here would be 9.88, right? I know I'm zooming through this, but I just want to get through this one problem and I'll let you guys leave. Okay, so it's essentially plugging these numbers in, right? Guys? So we plug them in, we get through this, you add them all up together, that's how you get 9.88. Again, this is our formula that we have. Then to use this chi-square table, you take the number, so first you have to find the degree of freedom. To find the degree of freedom, you take the number of genotype classes and minus that with 1, okay? So in this case, you have 4 genotypic classes, and so our R value would be 4 minus 1 equals 3. So you come here, you have degree freedom here. You look at where it's closest to. So in this case, our chi-square value was 9.88, right? So in this case, you go around where it's there, but never go higher than this value. So you go, okay, so we come here, we see 9.348, but we see 11.345, right? So in this case, you would go with 9.348, which gives you a 2.5 or approximate p-value of 0.025, okay? So again, the standard cutoff for this, unless they state otherwise, 5% is your cutoff. So in this case, since the p-value is lower than the standard, this hypothesis of no significant differences would be rejected. This means that there is the difference between the measured values and the predictive values are significant. Does that make sense?
Yeah.
I'll put up a video for more of these kind of questions too, so you guys can like look at it and prepare for your exam that's coming up in lecture. Does that make sense?
Yes.
Okay, so the next part of this involves pedigrees. It's also in my genetics, and not genetics, in the discussion videos that I have for you guys, that you guys take for the quiz. So definitely look at that, and I will let you guys leave because I am already overtime right now. So
The
I
quiz will
hope
be later tonight.
it will be up later tonight, yes.
Okay, thank you.
Is the
Yep.
participation assignment on Blackboard?
Wait, say that again?
Is the participation assignment on Blackboard?
Okay, since I'm running overtime, everyone just write, type in here in the group, in the Zoom chat, like now. Wait, did I lose someone before? Because I think. But yeah, after you guys are done writing here in the group chat, the Zoom chat, you guys are free to leave.
Thank you.
Yep.
Thank
Have a
you.
good day.